In this week's lab, we are going to be doing two things. We're going to be looking at osmosis and diffusion. In the first part of the lab, we are going to be looking at osmosis. We are going to cut potatoes into tiny cubes and put them into three different types of solutions. One solution is going to be a hypertonic solution. Now this is important to osmosis because osmosis is the movement of water in and out of a cell. In a hypertonic solution, there is more solute in the solution, and what's going to happen is the water in the solution has lower free energy than the water in the cell. So water from the cell will move out into the solution. We can expect that the potato in that solution will get smaller. The next one will be an isotonic solution. In an isotonic solution, there's an equal amount of solute outside in the solution as there would be things inside the cell. So water has an equal free energy in the cell and in the solution outside, so water will move both ways equally. And the last one is a hypotonic solution where there is less solute outside the cell than there are things inside the cell. This means the water has a higher free energy outside the cell, and water will move from the solution into the cell, and we can expect those potato cubes to grow in size. In the second part of the lab, we are going to measure diffusion. This is not when water is moving, but this is when molecules are going to be moving. Now, molecules can move across a cell membrane based on three different factors. First off, their size. Larger molecules move across a membrane slower. Second off, their polarity. More polar molecules will move across a membrane more slowly because the membrane has a hydrophobic or nonpolar interior. And third, the presence or absence of transport proteins. If there is a protein that can let the molecule through, it will move through very quickly. We are going to measure the movement of molecules into cells indirectly by looking at how the molecules that we choose, such as urea, propanol, ethanol, and various other ones, are going to move into the cell, because as a molecule moves into the cell, this increases the concentration of solute in the cell, and water will move from where it has higher free energy, which is now the solution, into the cell. We are going to watch the cells expand as molecules move into them. So how fast the molecule moves in is how fast water will move in, and since we will be using red blood cells, they will explode when the water moves in. So we are going to pipette some red blood cells into a tube, and we are going to use this black box with a string and the tube in front of it. You can see the string through the tube. That means the tube is clear. When we have red blood cells in the tube, when the blood cells are whole, it will look like this. You cannot see the little line through the tube. When the cells have lysed, you are able to see the black line through the tube. So this is how we are going to use a pipetter, a test tube, some red blood cells, and this black box to measure the lysis of cells. You're going to put your flashlight through this, if you have a flashlight on your cell phone, and that will help you to see things. We are going to time how long it takes for the cells to lyse, and know that the longer they take to lyse, the slower the molecule is moving into them.